So this video is going to go through some of the what are often called exceptions to Mendel's rules, but are really just additions to Mendel's rules. Some things we have learned since Mendel's time, now that we understand about genetics and chromosomes and other patterns of inheritance that don't follow strictly Mendel's laws of dominance and recessiveness, and give us the exact proportions of predicted offspring that Mendel's laws do. So essentially, there are some genes that act a little differently than standard Mendelian traits, and that's what we're going to talk about here. So some genes have more than just two alleles. Up till now, the traits that we've looked at had like two versions of the genes. So there were either purple flowers or white flowers, or there were yellow peas or green peas. But for a lot of genes, there actually are more than two alleles. So there's more than two options. That doesn't really change much of what we do with them because every organism still only gets the one, well, the one chromosome from each of its parents, so the two total alleles. But there are just more options in the population. So in terms of like doing Punnett squares or anything, the rules for genotype and phenotype are still the same. The one difference here is that this is where you might get a hierarchy of dominance. A hierarchy of dominance. So you might have, like, in this case here, we're looking at rabbit fur color, where you have the wild type, which is dominant all the time. It's dominant over everything. Then you have this chinchilla color, which is like the black-tipped white fur, so you get a gray colored. And that's recessive to the brown fur, but dominant to the Himalayan pattern, which is white fur with just dark points. So the allele colors here, it's a lowercase c with a ch is how we indicate the chinchilla pattern. And that's going to be dominant to the c with a little h, which is the Himalayan pattern. And then there's also the white fur albino, which is the most recessive of all. So you sort of have a hierarchy. Big C is dominant over chinchilla. Chinchilla is dominant over Himalayan, and Himalayan is dominant over white. So you have sort of this series of rules. And so when we're interpreting phenotype, that can have an effect. But when we're looking at actually doing the genotype and like doing a Punnett square with these, we do it the same way we've done it before, right? So if we have our parents that we're going to cross, say we have a brown rabbit, so big C, that we're going to cross with a, let's say a heterozygous chinchilla rabbit that is heterozygous with a Himalaya gene as well. So this rabbit would look, the phenotype, it would look like a chinchilla colored rabbit, so it would be gray because the chinchilla color gene is dominant over the Himalayan color, even though they're both recessive to the brown wild type. And so we're going to do this the same way that we would do a Punnett square before. We're going to look at the possible gametes. So anything with a big C, a homozygous individual is only going to be able to pass on a big C allele. So it's going to pass on two big C's. Whereas our heterozygote here, this one's going to be able to pass on either the chinchilla gene or the Himalaya gene. So then we take those and we're going to put the dads across the top, so big C, and the moms across the bottom, little c, ch, little ch. And the Punnett square is going to just help us organize what their possible outcomes could be. So we're going to cross them. So this one, big C, could combine with the little c, ch. And this big C could combine with that little c, ch. This one, big C, could combine with the little ch. And this big C could combine with that little ch. So we get this sort of 50-50 Punnett square. 50% of the offspring would be big C, little c, ch. And 50% would be big C, little ch. Now, those are the genotypes. That's the list of alleles, but what would their phenotypes be? Well, in this case, we've got the big C, which is brown, and the recessive chinchilla color, but because we have that dominant C, the rabbit's going to be brown. And the same is actually true down here, that you have that big C and then the little c Himalaya, But because it has that big C, it's still going to be brown because that's the dominant trait. So in terms of phenotype, it's 100% brown offspring would come from this crossing. So this is our phenotype. Our phenotype ratio would be 100% brown. Our genotype ratio would be 50% heterozygous with chinchilla and brown, and 50% heterozygous with Himalaya and brown. But as you can see, doing the Punnett squares with this multiple allele situation is the same as before. It just means that there are more options out there in the population. Each individual still only gets 2 copies of the gene. So like there's no rabbit out there that has a brown gene and a chinchilla gene and a Himalaya gene. They have to just get the 2 traits. And that's because they only have 2 of each chromosome. So then if we move on, another kind of exception or addition to Mendel's laws are that some genes have more than one dominant trait. So more than one trait is actually dominant. This we call codominant. We have codominant traits that share dominance. And what this produces is that you have multiple different phenotypes. You have 4 or sometimes more, depending on how many different codominant genes you have, but you have at least 4 phenotypes. They can be all the first dominant, all the second dominant. They can have both dominants, so they can display both dominant traits, or they can be the recessive trait. So those are your 4 options. One of the most common examples of this is looking at blood type in humans. There are 3 alleles for blood type in humans. You can be the genotype, what we say show it with an I because it's referring to plasma components in the blood, and we say you can be IA. That's the one gene, and that contributes to A-type carbohydrates on the blood cells. You can have the gene IB, and that creates B-type carbohydrates on the blood cells. Or you can be IO, which means you have no carbs on your red blood cells. You have neither A nor B type carbohydrates on your blood cells. So those are your 3 alleles. Those are the 3 things that you can inherit. But A and B are codominant, which is how we get blood types AB, because those are people who have the IA gene and the IB gene. So they have both of them. So both of them get expressed because they are both dominant. They're codominant. So they have A carbohydrates on their blood and B carbohydrates on their blood. And we can see that in the chart when we look at AB type blood. They don't make antibodies to those A and B carbohydrates because they are present on their blood. We only make antibodies to things that aren't on our blood. So again, if we were going to do a Punnett square like this, it operates the same. The rules of Punnett squares are the same. The difference is how we interpret the heterozygotes. Because of codominance, if you have both A and B, then both are expressed. And you can see an example here with this flower. It is codominant with red and white. So it has the red gene and the white gene. And so some of its petals are white and some of its petals are red. Note, none of the petals are pink. That's actually a different sort of inheritance pattern that we're going to talk about next. So in codominant situations, the heterozygote that gets both of the dominant genes expresses both of those genes fully. You get the A-type blood and the B-type blood or carbohydrate on our blood. You don't get some like mixture of the A and B carbohydrates. You just get the A and the B both expressed. Like here in this flower, you have the red parts and the white parts. You don't have any pink. However, some genes actually do exhibit a little bit of blending characteristics, and when that happens, it's because there's not really a dominant allele, and this is what we call incomplete dominance. It's incomplete dominance because none of the genes are actually dominant. So in this case, you only get 3 phenotypes, rather than in the previous you got 4. You can have all one extreme, all one genotype extreme, all the other extreme, the other end of the spectrum, and then you get a blending. You get something that's somewhere in the middle. So, for example, there are some flowers that breed this way with incomplete dominance, so that you have red flowers when they are homozygous, sort of, for one allele. You have white flowers when they're homozygous for the other allele. And then when they're heterozygous, you get pink flowers. So the heterozygote is actually a blend or a mixture of the two phenotypes, the two other phenotypes. So again, this is just going to be a difference in how we interpret the heterozygote trait. We're going to do our Punnett squares exactly the same way. For our flower example that we have here, we would have your big A, and your little a. Well, these guys can only make big A gametes. This one can only make little a gametes. So when we go to do our Punnett square, we'd put two big A's there, two little a's there, big A, little a, big A, little a, big A, little a, and big A, little a. So their offspring would be all heterozygotes, but rather than these heterozygotes all being red, like the big A would show, because this is incomplete dominance, these would be 100% pink for their phenotype ratio. Pink, because of that incomplete dominance, is going to give you a blend of the red and the white. And of course, the picture here is showing if you were to breed these pink flowers with each other, now you can have a big A or a little a on both sides. So we do that Punnett square, and we have big A, little a, big A, little a. Here you're going to have big A, big A, big A, little a, big A, little a, and little a, little a. This one would be red. This one would be white. But these two would be pink because they're that heterozygotes with incomplete dominance. They're going to show that blending. Now I have a picture here just to remind you that incomplete dominance and codominance are not the same thing, right? Incomplete dominance is where you have a blending of the two different colors. Codominance is where you have distinct parts expressing both of those dominant traits fully. So like you get this sort of blocking pattern on the snake rather than this blending pattern on the snake. They are two different, similar, but different exceptions to Mendel. It's very important that we understand how they are different. One of the last real big additions to Mendel that we're going to talk about are what we call regulatory genes. And here we are back talking about cats for this example. Regulatory genes are genes that regulate the expression of other genes, or in other words, they control the expression of other genes. A lot of times, these regulatory genes are what we call silencing genes. So when they're present in a certain allele, the certain genotype, they actually cause other genes not to be expressed. So for example, we've probably all seen pictures. If not, there are cats that exist who have no tails, right? Sort of like bobcats. There are domesticated cats that have no tails, the most common example of which is the Manx cat. And most cats have tails, and they have genes that are associated with their tails, such as how long it's going to be, how agile it's going to be, what color pattern is going to be on it. But there's another gene that just controls whether or not there is a tail. And in this case, the dominant version means no tail. So the big S is no tail. And the recessive version, which most cats have, is tail. Because remember, just because something's dominant doesn't mean that it's the most common, right? In this case, the recessive gene is actually the most common. Most cats have tails, but if they get this dominant gene, it's going to make them have no tail. And so that is a regulatory gene. It doesn't matter what other traits they have, what other genes they have, and they will have other genes that will say, oh, we're going to have a curly tail. We're going to have a tail with long fur. So it might have the genes that code for curly tail and the genes that code for long fur tail. But if it has that big S, there's going to be no tail, which means these other genes aren't going to get expressed. It doesn't matter that they have the gene for a curly tail or for a long fur tail because they're not going to have a tail. So that gene is really important. And so at the end of this video, I would like you to work out this question. Can two Manx cats without tails have a kitten with a tail? Use Punnett squares to back up your answer.